So we're going to talk about DNA replication, and there's a lot to get through here because it's kind of a complicated process, but it's pretty straightforward once you understand the basics of DNA structure. So, why do we need to go through DNA replication? If we're going to divide into two new cells during mitosis, we need to make sure each cell has the same amount of DNA. We want to make sure that we're making functional cells, not cells that end up with half the DNA that they need. So in order to repair, replace, grow, and develop, we want to make cells that are exactly identical to the one we started with. We call DNA replication semi-conservative. That means that we're going to use our original strand of DNA as a template and build the DNA strand directly onto that template. There are three main stages of replication. There's unwinding, base pairing, and joining. Unwinding consists of the enzyme helicase coming in and unwinding and unzipping the DNA. Remember that unwinding is where we untwist that twisted ladder or that spiral staircase. We're unwinding it. We're making it straight. Unzipping means we're breaking the hydrogen bonds that hold the base pairs together. The DNA is kept open by a set of proteins called the single-stranded binding proteins. As the helix unwinds, another enzyme called the RNA primase adds a short segment of RNA called an RNA primer to each strand of DNA. And that primer kind of acts as like a flag of, hey, you're working from here and moving forward. So this is what that looks like. So we have our helicase coming through and unzipping our DNA. We have primase adding on our little RNA primer. You can tell that it's different because it's got a different color. So the DNA is here represented as blue and the RNA primer is red, of course. And then we have these single-stranded binding proteins which are holding open the DNA, preventing it from zipping back up. Base pairing. So here we have an enzyme called DNA polymerase. DNA polymerase is the guy that's going to be doing most of the work here. He comes in and makes sure that the matching nucleotides all get sealed up together and kind of adds those new nucleotides to the DNA so that we're forming the strand as we go along. Our DNA polymerase can only move in one direction, however. It can't move in the two different directions that DNA runs. Remember, DNA is anti-parallel, which means one side of the DNA is running in one direction and the other side is running in the opposite direction. And DNA polymerase cannot work in that opposite direction. It can only build strands 5' to 3'. And I'm going to talk a little bit about what 5' and to 3' means. So we're going to have to build the strands in opposite directions. Because of that, one side is built in one continuous piece, and we call that the leading strand. So we are calling the two different strands, we're calling them two different names. So the leading strand is the one built in a continuous piece. The lagging strand is built in large segments. These segments are called Okazaki fragments. Okazaki fragments are later put together by the enzyme DNA ligase. So we build these large segments, these large fragments, and then DNA ligase comes through and makes sure they're all linked together so that we end up with two strands of DNA. So I said I was going to make kind of clear up what 5' to 3' is. And what 5' is referring to is the number of the carbon. So we can number the carbons based on their position. So we have a nitrogen-containing base paired up here to this carbon. And so we call that carbon number 1. That's 1'. And then we have 2 just going down straight from that one, then 3, then 4, and then our 5th carbon is actually up here connected to the phosphate group. So what we're saying is that we have to go from 5' to 3', so we can only build in this direction. This strand has to be built in the opposite direction. So this is what this looks like. So we have DNA polymerase coming through, and it's building our strand through 5' to 3'. And then we have the lagging strand on this side, where we just have these RNA primers that have segregated up the strand, and then we're building in these short fragments to show you how this gets built. And then, of course, we have DNA ligase here linking the strands together. And where we're moving is we're trying to go from right to left here in this picture. And so we're moving in this direction, but we have to build in the opposite direction on the lagging strand. Then finally, we have joining. Joining is when DNA polymerase comes into contact with the RNA primer on DNA, and it removes the primer and fills in the space with DNA nucleotides. When the RNA primer has been replaced, DNA ligase links the 2 sections. It is important to note that DNA replication does not occur starting at one end of DNA and working to the other. DNA replication occurs in many places on the DNA strand all at once. We call these the replication bubbles, and they're segments of the DNA strand that are being built, and they start all down the DNA strand. And as replication continues, they start getting larger and larger, and eventually they join up and they become 2 solid strands of DNA. In DNA, we call the new strand the daughter strand. In this picture, it's the red strand here. And the blue strand is called the parent strand or the template strand. And if you can identify the problems with this center picture, you probably remember our previous picture where it's not just some jumble. We can build one side continuously and the other one has to be built in fragments. I'm going to link this video into your notes that you're so that you can see it in your Nearpod, so you can watch it there. And then finally, we have the terms that you need to know. You need to know what semiconservative replication is. You need to know what the parent or template strand is. You need to know helicase, single-stranded binding proteins, RNA primase, RNA primer, DNA polymerase, leading strand, lagging strand, Okazaki fragments, DNA ligase, replication bubbles, and the daughter strand. If these terms are missing from your notes, please rewind the video to where I'm talking about that item and watch the notes again and get these into your notes.